So our experiment was pretty much the same as the one that we did in class with the spinach and the syringes and measuring the level of O2 production. Except the main difference, while everything else was basically the same, the main difference of ours were that we were testing different sources of light uh, and its effects on the production of O2. So we tested UV light, we tested an iPhone flashlight, we tested a lamp light, and we tested a control group, which was no light. So um, we hypothesized that the one with the UV light would produce more O2 because it has a higher frequency, so therefore it would emit more energy, and we thought that would basically um, power the photosynthesis reaction and make it produce more O2. So the design of our experiment was basically we had the same procedure as the previous one. So we basically would hole punch the spinach, and we would take 10 sections per uh, light. So we would basically have 40 hole punches in total, spread out over 4 groups, and one being the control. And then we would basically suction the spinach sections inside the syringe, which would basically seal it with the sodium bicarbonate with soap. So that would basically create a suction around it, and that would basically trap the O2 as it's being produced when we were experimenting. And we basically made each cup with the fluid with the height of 3 centimeters, which is basically eliminating any variables that would happen, or anything that would change the experiment if the level of fluid or sodium bicarbonate was higher. So that basically was just another control. And we tested each one under a duration of around 15 to 20 minutes. And we recorded the level of O2 through measuring how many of the sections were floating. So basically, every 30-second interval, we would have a stopwatch on our phones, and then we would time it. And if a certain amount of sections were floating in that time period, we'd mark it. So then we basically had our data being 30-second intervals with the level or the amount of slices floating. So as you can see in the image here, nothing really changed for our first three, which were the iPhone flashlight, the UV light, and the no light. So nothing was floating. However, in our last experiment, we found that the lamplight had actually let some of them float. So that was basically our only successful one. So that basically kind of disproved our hypothesis. So every other plant, as you can see in the graph right here, just didn't work except for the plant one. So as a result, our conclusion was false.